All right, welcome back, guys. We have some more content to cover regarding the nervous system. So we're going to continue progressing forward with this stuff. Let's see where we left off. So where we left off, we were talking about the neuron. We were identifying what the neuron looks like and its basic structures. In this video, we get to learn a little bit more of the structures that make up the neuron, and then also go a little bit further into some more material overall. Okay, so first of all, the nervous cells have a lot of supporting actors, right? So these are all tissue that help out. So first of all, the supporting cells are more referred to as, your neuroglia. That is their name that we have. Now, there are a number of them. If you notice the central nervous system, there are 4, and peripheral nervous system, there are 2. Why would central nervous system have so many more? Well, simply, it's because the central nervous system is the brain and the spinal cord. It's going to need more assistance. Now, we don't need to know all of these, but the ones that we're going to pay attention to are oligodendrocytes and Schwann cells. Those are the ones that we're going to spend more time with. The oligodendrocytes and Schwann cells you'll see on the next slide, but let's talk about these others as far as what they do, like no particular order. So astrocytes, microglia, ependymal cells, and those oligodendrocytes, they produce a barrier between the capillaries and neurons, protect the neurons, provide some sort of immunity, line the brain and spinal cord cavities, wrap the nerve fibers, and most importantly, which is especially these guys, produce myelin sheath, which we'll talk about in just a bit. The Schwann cells are going to help, and satellite cells are going to surround large neurons and offer protection and cushioning. Here we see what a lot of those cells look like, right? You see your astrocytes, microglial, ependymal cells. These 2 guys right here are the ones we need to pay more attention to: oligodendrocytes and satellite cells. The big thing that you need to process, or what we need to comprehend, is that the oligodendrocytes are essential for making myelin, which we're about to talk about, and it makes myelin specifically for the central nervous system. Satellite cells and Schwann cells, but we focus most of our efforts on Schwann cells, make the myelin, and more specifically, make the myelin for the peripheral nervous system. So I do have a mini mnemonic for this. I had a student ask me, hey, is there a way to remember this? Because this would be like a 50-50 question on a test. What I think helps is when I see the word Schwann, named after the scientist who discovered this. To me, Schwann sounds like very formal and regal almost. So I think of like a really fancy bougie pen. So I think of a Schwann pen, and I just happen to spell it like this, or pens. I underline the E because the E is not necessary for the mnemonic, right? Or the not necessary for what we're trying to remember. But Schwann cells make myelin for the PNS, peripheral nervous system. So that kind of works for me. If it doesn't really work for you, not a big deal. That's the whole intention of these mnemonics anyway. Okay. Next, we can talk about the myelin. What is it? So if you had a powerful microscope and you're looking at a neuron, on the axon, you would see all these white little regions, right? All these white little regions, all these pods. Those pods are made up of a compound called myelin. Myelin is this whitish fatty material that covers the nerve fibers to speed up nerve impulses. I think of like Mario Kart, where if you're driving and you run over those little arrows on the ground and it speeds up your character and gives you a boost, that's a similar process as to what's happening here with myelin. It's boosting the impulse as far as the speed of it is concerned. Now, what makes the myelin, especially in the PNS, so let's just make sure we are observant of that. What makes the myelin is Schwann cells. Literally, the cell itself is sitting on the axon and it's making all this extra layer of stuff. Now, sometimes you saw me earlier say myelin sheath. The myelin sheath simply is just layers of myelin. So nothing too complicated there. Now, this is interesting. You'll see how there's gaps, right, in between each myelinated region. If we just said that myelin speeds up the signal, why would you have gaps? Well, we're going to learn this in the next video more in detail, but these areas are known as the nodes of Ranvier. Once again, named after the individual. But what's fascinating is that when you receive an electrical impulse, and we talked about this in the last video, we mentioned how when you receive an electrical impulse, it's going to travel in the direction of the dendrites, through the cell body, and then eventually when it reaches the actual axon, what we observe, it's a concept called saltatory conduction. What we observe is that the impulse is going to jump from every single one of these myelinated regions as it progresses through. That jumping process speeds up the signal even faster. What's fascinating is that for nerves to do their job and neurons to send signals, and for your brain to comprehend everything that you need to do on a daily basis with no delay, it's got to work at a speed of milliseconds. And if you're not familiar, a millisecond is 1/1000th of a second. So a lot of stuff's happening very, very fast. Some more stuff here we need to talk about. The ganglia, I don't get too crazy with ganglia. It's just a collection of cell bodies. It's just a definition if you need to pay attention to that. If you were talking to like a medical professional, like a neurologist, there is some technical terms that you'd use. So, for example, bundles of nerve fibers is considered a tract in the central nervous system, and bundles of nerve fibers is considered just nerves, the peripheral nervous system. I tend to just use the word nerves in general sense. White matter and gray matter. We do need to talk about these. White matter is defined as dense collections of myelinated fibers, so it's areas that are myelinated, whereas gray matter are areas that are unmyelinated. If you paid attention to here, this is the brain. The spinal cord is very similar as far as white and gray matter. I would estimate, this is nowhere near like an actual prediction, but I would estimate that roughly 55% of the brain is white matter and roughly 45% of the brain is gray matter. Why do we see that though? Because white matter, we said, is myelinated. It makes sense. Myelin is for quick action, right? So why would you have areas of the brain that have no myelin? Well, what it turns out, and we're going to learn this more in nervous system part 2. Sorry about that. What it turns out is that you have regions of your brain that they're literally, their whole job is communication, communicating between both hemispheres, to be more precise, left and right hemisphere. So that white matter is more ideal for communicating information. Gray matter, on the other hand, is more ideal for integrating, processing, calculating, figuring things out, right? So you need both. You need both for your brain to work properly. Functional classification. So there are different types of neurons. I only pay attention to the different ones that do different jobs. I'm not going to worry about learning or having you guys learn too much about the structure of these different neurons, but we'll talk about it briefly as we conclude today's video. Knowing that there are 3 different shapes or different functions of neurons, let's go over that real quick. The key thing here, honestly, is just pay attention to the first word or the prefix. So look at sensory neurons. What do we think sensory neurons are associated with? Your senses. They're associated with your senses, and they're more specifically carrying sensory information to the central nervous system. So they're sending information towards the central nervous system. Motor neurons, on the other hand, are going to send signals away from the central nervous system. So these guys are going to send signals away from the central nervous system to activate something, be it muscle, a gland, whatever's in its path. Interneurons, these are kind of tricky because they don't have a name to kind of depict it. But if you look at the prefix, inter reminds me of in between, especially when you look at the definition. They are connecting sensory and motor neurons together. That's their entire job. So as long as you can recognize this, I think this wouldn't be too problematic for anyone, right? Here we see a little bit more about these types of neurons, and you can actually see up here what we're about to preview as far as the shape of the neuron. I'm not going to worry too much about this stuff as far as learning the shapes of the neurons because literally 99% of the neurons, 99% of the neurons are multipolar. So that's what we're going to spend most of our time with, right? That does not mean bipolar and unipolar neurons shouldn't be learned. That's something that you can learn on your own time, especially if you are pursuing this stuff further in the future. But for now, multipolar ones are the ones we spend most of our time with, especially because this is the guy we've been learning. This is the typical neuron that we expect to see. These other neurons, like I said, are relevant. They have other jobs that are necessary, but we don't need to dive too deep into that. Okay, so that's what we're going to conclude for today. I think that's a great place to stop. Next video, we're really going to get into action potential and learning more about that process, which can be a little bit more dense. All right, so I appreciate you guys checking in. I'll see you guys in the next one.